Hello there, my wonderful biology students. Welcome to 1.4, membrane transport. The membrane never, ever ends. No. Okay, so in the last section, we learned about like what the different models of the cell membrane have been, how they were disputed and moved on, and then we learned about the different parts of the cell membrane. And now we're going to learn about some transports, right? So we talked about sometimes there's passive transport, active transport. There's the proteins that have certain signals. So how do we get things in and out of the cell, right? So how do we get things that we need into the cell, things that we need out of the cell, and so on and so forth. We're going to start with like the simplest type of transport, which is passive. And we will be doing a lab with osmosis. Yay. All right. So in order to do our lab with osmosis, first of all, we have to know what it is, and then how we're going to design an experiment to actually quantify the results to be able to measure the amounts of what we're going to be looking at water going in and out of an object, okay, or across the cell membrane. All right, so let's talk with osmosis. Osmosis Jones, such a good movie. Have you seen it? We should watch it. But it really has nothing to do with osmosis, except for that, big fancy science word. But osmosis has is a special type of diffusion having to do with water. Sorry, I'm moving my mat over here. Osmosis Jones is a white blood cell. He polices the body of Frank, who gets sick. So not this guy. Okay, so whatever knowledge you have of osmosis Jones, right? We're looking at diffusion of water, right? So in osmosis, we're looking just at water. Everything else is a type of transport. In this one, we're just looking at water. Diffusion is a word that's just kind of thrown around, so I want to make sure we understand what that is. But you probably have known, what it means to diffuse a situation, to like break apart. If you have a diffuser at home, how it spread things apart. And so diffusion, is a type of passive transport, and where the concentration of molecules or particles in a solution keeps moving until it's even, right? So in this case, we have like a little drop being here, it's going to move around, it's going to move until it all spread around evenly, concentration. So what does that look like? So this process right here does not require energy, right? So diffusion is a passive transport that does not require energy. So I have this kind of set up for you guys right here. If this is my membrane here, and let's put a couple leakage channels here, right here, and here. I'll put two of each, okay? And I have inside and outside my membrane. I'm going to start, all right, let's put a bunch of these. This one's here. Let's put a bunch of these on this side, right? So what I'm going to see with my concentrations, right, is that when I have these leakage channels open, right, that I'm going to get that the particles moving to different sides of the membrane until they are equal, right? So my leakage channels allow us to do that. You also have gated channels, right, which can open and close, and those happen in certain situations when needed, right? So if I wanted to open those up to kind of make that faster, right? Let's say that there's something going wrong with the cell and I need to even those out evenly. Your gated channels might open up, and then when it's even, they can close again. Okay, so that's the type of diffusion, is when it goes back and forth against a membrane until it is equal on both sides. Okay, all right, so let's go back to passive transport. Passive transport is when I'm going against a concentration gradient, so I'm going to need energy. There's energy needed to get things across a membrane. Okay, that's the demo I just showed you. So passive, right, going back to osmosis is when things are moved from place to another without the use of energy, right? So it's a natural kind of movement, a natural type of flow. Osmosis is a type of passive energy. Passive transport, no energy. Active transport requires energy, right? So those are kind of easy to remember. But it's when I'm moving from one place to another and I actually need energy to do so, right? So in this case, it's going down, right? So there's not that energy. In this case, we're going up the stairs with the refrigerator, right? So active and passive. So now let's look at, so that's kind of like big idea, but let's look at kind of the nitty gritty details, because that just sounds too simple, right? It all has to do with what we call a concentration gradient, right? I like to kind of think of the word grades when I think of gradient, and I'll tell you why, kind of my mnemonic devices before it, right? So things, if you think about your grade This is my kind of analogy with gradients. If I'm going against the concentrated gradient, right? So I'm trying to go against it. So like move my grade up, it's going to require energy, right? If I'm going with the concentration gradient, right? So I'm just kind of going with them, I'm doing nothing, that's passive, right? So things can move against it, it can move along it, it can go down, or it can move within it, right? These last three things all kind of mean the same thing. So along or down or within all mean the same thing, right? If I'm going against the gradient, that means that I'm going like, I'm going to need some energy because I'm going against what kind of the cell wants to be doing. Okay. You can also have electrochemical gradients, lots of different type of gradients. Of course, we'll be talking about those fun things. Okay. All right. So what is exactly a concentrated gradient is not your grades, right? But it's when there's more of something on one side of the membrane than the other, right? So if I go back to that, that simulation I was showing, if I had like a bunch of the, green on one side and there's only 2 green on this side, like 10 and 2. So if I leave it alone, the concentration will balance out over time. It reaches equilibrium. So if I let those little like green bounce around a much, over time, they would balance out. But sometimes we need to get it against our concentration gradient, right? So sometimes I need more of something inside or outside of the cell. And the cell membrane is like, remember that bouncer, that thing that decides what the levels are and what needs to happen for the cell, what needs to come in, what needs to go out, what channels need to open, what channels need to close to make those that concentration gradient what it should be. Okay, so here's our 2 different types of transport. We're going to be talking about endo and exocytosis in our next lecture. But passive up here, here's the 3 different types of passive that we have. So osmosis is with water, right? Diffusion is going to be anything that's dissolved in a liquid or a gaseous form. And facilitated diffusion are things that are too big to fit through membranes, but none of these are going to require energy, right? So they all move along the concentration gradient. In this case, for facilitated, we get proteins being used. Okay. Now for active though, we're going to need energy, right? So make sure you're looking at these things right here, right? So energy, all of these, no, right? Again, if I'm passive with my grades, just going to go down, right? No energy required, right? These all do require energy here. And these ones, right? It says across the cell membrane. This is my not always one. We'll talk a little bit more of that in class. But in this one, right? Against, along or against, depending on what is needed. Requires energy and doesn't are my big things, my differences there. Okay, so take a second to pause this. Make sure you have everything written down. In our next, here, let me see. There we go. In our next, what's it called? In lecture, we're going to talk about endo and exocytosis, and more particularly these this active type of transport. And then for our lab, we're going to be looking at osmosis, particularly, and how we can measure the movement of water across the membranes. I lied. I wanted to add these last couple of slides. So here's kind of a diagram for you to add there in your notes about what that would look like, right? So this is my diffusion. This is my facilitated diffusion, both passive, right? But this is using that transport channel. And then active transport is going to use the ATP. So we'll go back and cover those in just a second, but I did want you to have those 2 diagrams there. Looking at, again, I'm moving down the concentration gradient. So I'm going from high to low, H2O. That's how we osmosis. No? Okay. Freshman bio? All right. So going from high to low is diffusion. Going to high to low for diffusion with water is called osmosis. Okay. And then last thing here, here's one more kind of visual for osmosis, right? So in this case, it's getting a little bit trickier because I've got sugar on one side. Now in this case, we're looking at water moving around to equal out the concentration gradients. However, sugar cannot go through my membrane, right? So if I have a lot of sugar on one side, that's going to bring down the concentration of water, right? Because I'm looking at the ratio of my water to sugar molecules. So water in this case is actually going to move into the other side, even though this is going down lower, because there is no sugar until I get that same on the other side. So that's what we're going to be looking at more particularly in your lab. Make sure you have something like that written down, that sometimes when I have selectively permeable membranes, just the water can go across and the other molecules cannot. Okay. All right. Now we are finished. I'll see you guys later.